This is we are discussed about the microbial world. So these microorganisms hate us, especially COVID-19 is a huge problem. We are suffering from these diseases almost, I think, more than 1 hour. 1 hour. More than 1 year. Microbiology is the study of microorganisms that are too small and are not visible clearly to the naked eye or unaided eye when they exist individually. These organisms are referred to as organisms, microorganisms. So, microorganisms include bacteria, archaea, cyanobacteria, blue-green bacteria, fungi, and protists. Molecules such as mycoplasmas and photoplasmas, viruses, viroids, and prions are also studied under microbiology. So let's go to study microscopic nature of microorganisms. In general, microorganisms are less than 0.1 millimeter in size and cannot be observed with unaided eye. Therefore, they must be observed with a microscope. Microorganisms and their structural components are measured in micrometers and nanometers. 1 micrometer equal 6 to the power 10 meter, and 1 nanometer equal minus 9 10 to the power um, m. So some microorganisms that are more readily visible than other because of their larger size. So microorganisms are ubiquitous on Earth. They are found in water, soil, air, and interior and exterior surfaces of other organisms. Marine and freshwater microorganisms form the basis of food chain in oceans and freshwaters. Some of them do photosynthesis and are primary producers in aquatic environment. Soil microorganisms help recycling of chemical elements between soil, water, and air and living organisms. So microorganisms suspended in air as bioaerosols have the opportunity to travel long distance with the wind current and precipitate. Pathogenic bioaerosols cause opportunities for disease spreading. Only a minority of microorganisms that associate with other organisms, such as plants, animals, and humans, are pathogenic. A majority of them are advantageous or harmless. However, all viruses are harmful to microorganisms they attach to. Some microorganisms are capable of inhabiting extreme environmental conditions that are unfavorable or even lethal for other organisms. So such microorganisms are known as extremophiles. Extremophiles have been found inside the earth crust, deep sea at a higher pressure, extreme acidic or extreme basic conditions, hydrothermal wind, frozen seawater, and aerobic and anaerobic conditions. So extremophiles are classified according to the conditions in which they grow, such as extremophils. Their living condition is high temperature. Then psychrophils, their living condition is low temperature. Acidophils, their living temperature in acidic pH level. Alkalophils in basic pH conditions. Halophiles require sodium chloride, and barophiles lives in high pressure. Some of these extreme environment consist of more than one extreme condition. For example, many hot springs are acidic or alkaline in nature at the same time. So the deep sea are cold and remain in high pressure. Microorganisms live in such environments are adapted to live with more than one extreme condition. So one thing is that high growth rate of microorganisms. Rate of growth and reproduction of microorganisms are high. Microorganisms possess a high surface area volume ratio due to their smaller size. This means that they have larger surface areas available for exchange of materials from the external environment. As a result, flowing rate of microorganism materials into the inside of cells and the exit of waste materials to the outside of cells increase and results in high metabolic rate. Therefore, average generational time or the time required to double the population of microorganisms are relatively less. Then we are going to talk about morphological, nutritional, and physiological diversity of microorganisms. Microorganisms possess a diverse morphological forms. Bacteria possess diversity in their shapes. Basically, three distinct shapes: rod shape, bacillus, spiral shape or coccus, and spiral shape or spirillum. The coccus bacteria may arrange in different forms: coccus, monococcus, diplococcus, streptococcus, staphylococcus, tetrahed, and sarcinae. Bacillus bacteria may arrange into either diplobacillus or streptobacillus. Spiral bacteria may arrange into their either Vibrio or Spirella or spirochete. The cyanobacteria exhibit a great variety of shapes and arrangement, unicellular to long multicellular filaments. Multicellular cyanobacteria may appear as either filamentous or non-filamentous. Filamentous appear as chains and the non-filamentous appear as clusters or colonies forming spherical, cubical, square or irregular shape. Two morphological varieties are found in viruses based on their symmetry or of protein codes that are called icosahedral and helical. In fungi, some of them are unicellular and other multicellular. Consider mass of fine tubular branching threads known as hyphae collectively from mycelium. The hyphae may be septate or aseptate. Prions are smaller proteinous particles. Unicellular protists possess a wide range of morphological diversity, molecules, and pleomorphic or variable shapes. Microorganisms show a variety, diversity of nutritional types based on the sources of carbon and energy. Nutritional types of microbes are classified. There are four major nutritional types in among microorganisms: chemoautotrophs, chemoheterotrophs, photoautotrophs, and photoheterotrophs. Based on the utilization of oxygen, microorganisms can be classified to four physiological groups: obligate aerobes, obligates anaerobes, facultative anaerobes, and facultative anaerobes and microaerophils. Some microbes capable of fixing atmospheric molecular nitrogen show physiological diversity, free-living nitrogen fixing microbes and symbiotic nitrogen fixing microbes. So then we are going to talk about the types of microorganisms. The first one is bacteria. Bacteria, in singular bacterium, single-celled, unicellular, prokaryotic organisms. They show different morphological forms and arrangement. The most obvious structural feature of bacteria is the shape of individual cells. There are three basic shapes: spherical, coccus, rod shape bacillus, spiral shape spirally. During cell division, cells can remain attached to each other and forms different forms of cell arrangements. So now we are going to talk about the different forms of cell arrangement of coccus bacteria. Coccus cells divide in one plane. Divided cells detach from each other after cell division. The Diplococcus cells divide in one plane. Divided cells remain in pairs. So in Streptococcus, cells divide in one plane. Divided cells remain attached to chain-like pattern. Within tetrad rather, cells divide in two planes, producing 4 cells remain attached together. Then Sarcinae cells divide in 3 planes and remain attached in groups of 8 cells. And Staphylococcus cells divide in multiple planes and form the grape-like clusters. So then we are going to talk about the different forms of cell arrangement of Bacillus bacteria. So bacilli divide only across their shore axis. Therefore, there are few cell arrangement forms. Single bacillus, single rod shape. Diplobacillus remain in pairs after cell division. And streptobacillus occur in chains after cell division. So the third one, different forms of cell arrangement of spiral bacteria. Spiral bacteria have one or more twist. They are never straight. Vibrio is a bacteria, looks like curved rods. Spirillum, helical shapes like corkscrew and rigid body. And spirochete, helical shapes or flexible body. So bacteria show a diversity of nutritional types. 4 major nutritional types can be identified among bacteria. They are classified based on the source of energy and carbon. Now, first nutritional types is for autotrophs. The source of energy is light. Source of carbon is carbon dioxide, inorganic carbon. And examples are purple sulfur and green sulfur bacteria. Second nutritional type is photoheterotrophs. Their sources of energy is light, and the sources of carbon is organic carbon. And examples are purple non-sulfur bacteriums. So third one, chemoautotrophs. Their sources of energy is inorganic chemicals, and sources of carbon is carbon dioxide or inorganic carbon. The examples are Nitrobacter, Nitrosomonas, Theobacillus, Theoxidans. So the other types is chemoheterotrophs. The sources of energy is organic chemicals. The source of carbon is organic carbon. Examples are most of bacteria are examples of chemoheterotrophs. So according to microorganisms can be classified into 4 groups on the basis of their tolerance to oxygens. The first one is obligate anaerobic. These bacteria requires oxygen for their survival. They generate energy by oxidative phosphorylation. Examples are Acerobacter species. The second physiological group is obligate anaerobics. They cannot survive in presence of oxygen. These microorganisms generate energy by fermentation. The Clostridium is an example. So third one is facultative anaerobics. So these microorganisms prefer to grow in the presence of oxygen, producing energy by oxidative phosphorylation, but they can also grow in anaerobic environments using fermentation. So Escherichia coli or E. coli is an example. Then microaerophilic. These microorganisms can grow only in oxygen concentrations lower than those in air. Examples are Lactobacillus species. So some bacteria are able to fix atmospheric nitrogen. They show diversity in nitrogen fixation. Example, free-living nitrogen fixing bacteria, Acerobacter species, symbiotic nitrogen fixing bacteria, Rhizobium species with legume roots. So mostly bacteria undergo asexual reproduction by binary fission and in some occasions of fragmentation of buddings. In rare occasions, bacteria of 2 strains share a portion of genetic materials, though through the process of conjugation. So this is all about the microorganisms and not microorganisms, about bacteria.